Good morning. So I'm going to go ahead and get us all started. I am Dr. Nayana Blankson, and I direct the RISE program along with Dr. Bradley Brennan, who you'll hear from at the close of our session today. And also, I just want to acknowledge on the RISE team is Assistant Director Stephanie Bird and our Assistant Katana Woodruff. And so on behalf of Dr. Bradley Brennan and myself, we're excited to welcome you all today to the 2020 RISE Summer Research Symposium. And it's our first one that's virtual, but I think there's some positives about this virtual experience where we can invite others who would not have been able to come to campus. And so we're excited to welcome you all today, and we appreciate our mentors and guests who are with us today. And so just a little bit about RISE for those who are joining us for the first time today. The program's been funded at Spelman since 2000, and the purpose of the program is to address the underrepresentation of women, especially minority women, in sciences at the PhD level and beyond, and to produce the next generation of Black women scientists. In this current cycle of our RISE program, we introduced a new part of the program with the acronym FIRST, which is our Freshman Introduction to Research and Skills Training. And so we had 5 first-year students who were selected, and it's pretty competitive because we only have 5 slots. They were selected in the fall, and in the fall they attended modules and other sessions to introduce them to research. And then in the spring semester, they got into research labs for the first time. And we all know this spring was a little bit different because of COVID. And so they had a very unique experience. And then they also engaged in a summer program. And so today you'll hear presentations from those 5 students. Additionally, we have our upper class RISE scholars, and we also had some of them participate this summer with us in our summer program. And so you'll hear from the first scholars as well as our upper class, 4 upper class RISE scholars. And so on the screen you see the order of events for today. We'll have our presentations followed by at the end, we'll have a special presentation by some of our alumni. And so without any further ado, I'd like to just go ahead and introduce our first presenter, who is Tita Tambadou.
Hello, everyone. I can pull up my presentation at this time. So my name is Tita Tambadou, and the title of my presentation is A Strategy for Survival, the Influence of Gendered Racial Identity on Gendered Racism and Identity Shifting as a Coping Strategy among Black College Women. My research mentor this summer was Dr. Danielle Dickens, and she is an assistant professor in the Department of Psychology, and she is also the principal investigator of the Grace Lab. So I am a rising senior at Spelman College, and I'm currently studying psychology. I've been in the RISE program for the past 3 years, and I am also a research assistant in the Grace Lab. So if you've ever watched the film The Hate U Give, you'll remember that the main character Star is from a predominantly Black neighborhood, but she attends a white private high school. And you'll notice that in her interactions with her white peers, she tends to change her tone of voice, her language, and even her interests. So Black women in academic spaces tend to feel pressure to fit into normative expectations of language and physical attributes, such that they may choose to straighten their hair or refrain from using colloquial language. So Black college-age women often have less experience in navigating experiences of gendered and race-based discrimination in comparison to older Black women. And currently there is a need for more literature that examines the negative consequences of race and gender discrimination, such as identity shifting. There's also a need for more quantitative and more mixed methods research in this area, given that majority of current literature is qualitative. So the first variable in this research was gendered racial identity centrality, and this describes the extent to which being an African American woman is important to one's self-concept. So an example of this concept is that being a Black woman may influence one's social interactions and the groups that they choose to affiliate with. So gendered racial identity centrality has been found to serve as a protective factor against gendered racism. So gendered racism is oppression that targets one's race and their gender. And it is also known as a combination of sexism and racism. An example would be assuming Black women are hypersexual. The Pew Research Center recently announced that Black women often report being overlooked for important tasks in their workplace due to perceptions of incompetency and lack of intelligence, which are related to stereotypes perpetuated against Black women historically. Black women may choose to shift their identities in response to experiences of gendered racism. So identity shifting is the altering of one's language and cultural behaviors. So an example would be Black women may choose to straighten their hair for an interview because they believe that the straighter hair appears more professional, given that smooth hair is often associated with professionalism. So Black women may shift their identities to overcome barriers of gendered racism and also to cope with discrimination. Shifting can actually have negative effects on Black women's physical, emotional, and mental health. So there are two theories that we utilize to support our research. The first is intersectionality theory, which is an aspect of Black feminist theory, and it describes how social identities interact, influence, and naturalize one another in systems of privilege and oppression. So Black women's race and gender serve as important aspects in their self-concept. So the phenomenological variant of ethological systems theory was the second framework for this research, and it describes how one's understanding of societal expectations, stereotypes, and biases in their environment influences how they adjust to new and different social settings. So Black women's experiences in academia can influence how they interpret their role in representing Black women and also how they desire to be perceived. This research aimed to examine the influence of gendered racial identity centrality on gendered racism and identity shifting as a coping strategy among Black college-age women. The first hypothesis is that gendered racial centrality will be negatively related to identity shifting. And to explain, higher levels of gendered racial centrality will be negatively related to higher engagement in identity shifting. The second hypothesis is that greater reports of gendered racism will be positively related to greater levels of engagement in identity shifting. And the final hypothesis is that greater levels of gendered racism and lower levels of gendered racial centrality will be related to increased participation in identity shifting. Within this research, there were 129 participants, all of which were Black women attending an all-female HBCU in the South. The average age among our participants was 19, and there was a pretty even distribution across all classifications. The most common majors among our participants was psychology, biology, and political science. So this research utilized a survey design, and the convenient sampling method was used, given that we contacted participants via their college email. Within the email, there was a link to the survey, to the Qualtrics survey that included an informed consent form, then a demographic survey, 3 questionnaires assessing gendered racism, gender racial centrality, and identity shifting, and finally, participants were offered an incentive of $5 cash. The first questionnaire was African American Women Identity Shifting Scale, and this is a 13-item measure on a 4-point Likert scale. With a sample item, I have a different self at school than at home. The next scale was a gendered racism scale, and this was a 20-item measure on a 6-point Likert scale with the sample item, how many times have you been called a derogatory name as a Black woman? And next, the final scale was the gendered racial identity centrality scale, which is an 8-item measure and is on a 7-point Likert scale. And the sample item is, in general, being a Black woman is an important aspect of my self-image. So results of the Pearson correlation indicated that there was a positive relationship found between gendered racism and identity shifting, such that greater reports of gendered racism was related to higher engagement in identity shifting practices. However, there was not a significant relationship found between gendered racial centrality and gendered racism, and there was also not a significant relationship found between gendered racial centrality and identity shifting. Results of a linear regression indicated that gendered racism and gender racial centrality accounted for a significant amount of the variance in identity shifting, such that gendered racism and gendered racial centrality significantly explained identity shifting. And we also noted that gendered racism had a larger influence on identity shifting than gendered racial centrality. So the first results supported our initial hypothesis that Black women that have greater experiences of gendered racism may engage in identity shifting more. So this is consistent with literature that suggests that Black women shift their identities to cope with experiences of discrimination. The next finding was that Black women who report greater experiences of gendered racism and had a lower gender racial identity centrality tended to have higher levels of identity shifting. And supportive literature suggests that Black women that have a secure gender and racial identity tend to utilize healthier coping strategies and understand the risk of identity shifting. The first limitation of this research was social desirability, such that Black women that were less aware of their gender racial identity centrality may have felt inclined to select answers that appear more favorable. Also, the second limitation was that this research did not account for any changes over time or causation. And then lastly, participants in this study did not include older Black women or professional Black women. So a recommendation for future research is an examination of adaptive coping mechanisms, which are essentially positive strategies in response to social support or spirituality. Another recommendation for future studies is the examination of the psychological implications of identity shifting on Black women's mental health and self-esteem. And finally, a comparison can be conducted to examine identity shifting patterns among professional Black women and college-age Black women. So understanding the psychological impacts of the coping mechanisms used in response to gendered racism by Black women can have implications for mental health workers and also for educators. So mental health interventions can be developed to promote adaptive coping strategies among Black women in response to gendered racism. And also educators can increase representation of Black womanhood in their classrooms to foster a positive cultural identity development among their students that are Black females. So overall, I enjoyed my experience in the RISE Summer Research Program, and in the GRACE Lab, I feel as though I was able to strengthen my skills in scientific writing, research design, and statistical analysis. I also feel that this program was able to prepare me for graduate school, given that I attended enriching seminars on graduate school preparation, and I was also able to begin my personal statement. So I would like to acknowledge my mentor, Dr. Danielle Dickens of the GRACE Summer Research Program and of the GRACE Lab. And I would like to also acknowledge Dr. Naina Blankston and Mrs. Stephanie Bird of the RISE Summer Research Development Program. Thank you. These are my selected references. And thank you all for listening. All questions are welcome.
Thank you, Tita. We have a few minutes for questions from anyone. You can unmute yourself if you have a question. You can also type in the chat box.
I have a question, Tita.
Yes, Vanessa.
Could you kind of talk about your favorite part of your experience?
I think my favorite part of this research experience was getting to conduct different types of statistical analysis that I was not familiar with. So I was able to learn how to do a regression and a moderation and other types of analyses that were new to me. So I think that those, being able to have that experience will definitely help me for graduate school.
Cool. Thank you.
Any other questions?
I have a question. Great job, Tita. And just with this topic on gendered racism and identity shifting, what's something new and enriching that you learned about any of those concepts by doing this research over the summer?
So I think that I learned that identity shifting, it can be a subconscious process that we are doing that we don't even realize. And I wasn't aware before conducting this research that it has negative psychological implications and can cause stress among Black women. So I think it was very interesting to learn about other healthier and more positive coping mechanisms in response to experiences of gender racism as opposed to identity shifting.
Thank you. All right, our next presenter is Vanessa Eaton.
So good afternoon, everyone, or good morning, depending on where you are. My name is Vanessa Eaton, and today presenting the project titled Police and Policy Reform. So I'm currently a senior biology major at Spelman College, and this summer I was working with Dr. Howard Henderson, who's the director of the Center for Justice Research at Texas Southern University. So just jumping straight into the project, we've had a lot of different events come to surface recently, not just in America, but also globally. But particularly, we've seen the rise of the Black Lives Matter movement, and this was sparked by the death of George Floyd, who was brutally killed by a Minneapolis police officer. But he isn't the only one we've lost. We've also lost Breonna Taylor, Ahmaud Arbery, Eric Garner, Sandra Bland, Trayvon Martin, and Tamir Rice, who was only 12 years old when he was shot by a police officer. And I should emphasize that these aren't the only people we've lost. We've lost many more people, but these are just kind of the faces of the movement, and hopefully you recognize their names. And just in 2020 alone, police have killed nearly 600 people. And this hasn't just been happening recently. This has been an issue that's been occurring for a very long time, but we've had very few effective solutions to the problem. But President Barack Obama did try to address this issue during his administration. And on December 18th, 2014, he signed an executive order to create a task force that will address policing in communities and also try to rebuild their trust between those two forces. And about 5 months later, the task force released the 21st Century Report, and these included recommendations to do just that. And these recommendations were built around 6 central ideas or pillars. And these pillars are building trust and legitimacy, policy and oversight, technology and social media, community policing and crime reduction, training and education, and officer wellness and safety. And in this project, we're trying to determine what recommendations from the report are actually being implemented in police departments located in the United States. And so what does policing look like today? The Department of Justice conducted an investigation on the Baltimore City Police Department and found that they're violating multiple constitutional rights by doing the following: making unconstitutional stops, searches, and arrests, using enforcement strategies that produce severe and unjustified disparities in the rates of stops, searches, and arrests of African Americans, using excessive force, and retaliating against people engaging in constitutionally protected expression. And similar investigations were also conducted on the Chicago Police Department and the Ferguson Police Department, and they found similar results. On top is a quote from the Chicago Police Department investigation, and they found reasonable cause to believe that the Chicago Police Department has engaged in a pattern or practice of unreasonable force in violation of the Fourth Amendment. On the bottom is a quote from the Ferguson Police Department, and their investigation revealed a pattern or practice of unlawful conduct within the Ferguson Police Department that violates the First, Fourth, and 14th Amendments. So if well-known funded departments like the Chicago Police Department and the Ferguson Police Department are violating constitutional rights, then we can assume that they're likely not implementing recommendations from the 21st Century Report. And so now I'll kind of go into the anticipated methods and results. The project was not completed in my time at the Center for Justice Research, and so I'll kind of be talking about what the project would look like and the results that we would expect to see. And going straight into methods, first, we would select the most significant recommendations from the 21st Century Report. And this would be done by completing a literature review to identify what are the most commonly proposed and adopted metrics. And then we'd create an assessment instrument based on those selected recommendations. And these would ask questions like, how many minority officers do you currently employ? Or what are the most common type of complaints your department receives? And how many complaints has your department received? And then we generate an assessment rating to determine how well they demonstrate 21st century policing practices. And to ensure inter-rater reliability, we'll have at least 3 separate raters. Here in the table, I have an example of what an assessment score and rating would look like. Let's say this department received a score between 0 and 4, then they are performing below expectation. If they receive a score of 5, then they are just meeting expectations. And if they receive a score between 6 and 10, then they're performing above expectations. And for the departments that are below expectations or even just meeting expectations, we then provide action items to improve their assessment score, because we want them to be performing as an effective police department that's also engaging in safe practice. And as an additional measure, we'd also analyze the pattern of complaints received by the department in the last 10 years. And this is a good strategy to predict the assessment rating beforehand. And these complaints are important because that's what prompted the Chicago, Ferguson, and Baltimore investigations that I showed you guys previously. And civilians can complain about officers' alleged use and involvement in excessive force, sexual assault, intentional false arrest, theft, or the intentional fabrication of evidence resulting in a loss of liberty to another. And following the methodology, some of the results we expect to see, if the department received an increase in civilian complaints in the last 10 years, then they'll most likely perform below expectations on our assessment. If they received a consistent number of complaints in the last 10 years, then they will likely meet expectations. And if there is a decrease in complaints in the last 10 years, then they'll likely perform above expectations. And as for the discussion of future directions, the events that prompted the rise of the Black Lives Matter movement has a long history in the US. And again, this isn't just a recent issue. Policing has been something that's been violated and abused in the past and also currently now. So clearly there's an evident need for a change in police culture. And one solution is to revise policing policies. And Obama tried to do that in his administration by releasing the 21st Century Report. And that is an amazing resource that doesn't seem to be implemented in police departments, or doesn't seem to be something that they refer to. And so hopefully this project will resume at the Center for Justice Research, and there will be promising results that could actually be used to improve the policing practices of police departments in the United States. And my road to my PhD, I've been in the RISE program for 3 years now, and the most recent research I've done has been in the criminal justice realm, and that has been very intriguing and an engaging experience because I plan to pursue correctional psychology. So this research is something that I'm very proud to be a part of, especially now. It's really important that we have people who look like us, people who don't look like us, being involved in research. And policing is one of those things that we want a lot of people involved in because it's not just one type of person being policed, it's everyone. And so I'd like to acknowledge my mentor, Dr. Howard Henderson, and the Center for Justice Research at Texas Southern University, the Spelman College RISE program staff and students, and also the NIH for supporting my research and my audience as well. Here on the screen, I have me and my mentor's name and email if you'd like to ask me questions after the program. But I thank you guys for listening, and so I'll take any questions now.
Thank you, Vanessa. We have time for a few questions.
I have one. Great job. And my question is, based off of your research so far, what are your thoughts on this topic, or what does the research say in terms of community policing?
So definitely now it's kind of revealed an area that needs a lot of attention. On the news, you definitely see, like with the protests and how police are treating people and their response to communities speaking on topics like police brutality. It kind of seems like a really aggressive and drastic response. And so I definitely think there needs to be more research done, or just more involvement of people from every type of community to improve policing. I'm not just in like major cities where the protests are happening, but I think overall, because major cities like Chicago Police Department shouldn't have like violation of amendments in their investigation. And so I think definitely having more community involvement would improve policing and also build better trust.
Okay, thank you.
I have a question.
There's been a lot of discussion about the strength of police unions, and especially as they might be a barrier to implementing some of the recommendations in the reports, like the one that you mentioned. But what do you see from your research on this topic are some of the most significant barriers to police departments across our country implementing these recommendations?
So for one, I'm pretty sure you heard the phrase like defund the police. And I don't think we should just take away all the resources, because I think that is one issue, or one thing that's contributing to this issue. I think we need to actually pour more resources into different areas, like better training, better community involvement, things like that. So just, I think resource and funding is one of those barriers that's like really important, but directing it in the right way and not just pouring it into giving them more weapons and more gear and things like that.
Thank you.
Okay, well, thank you guys for listening, and hopefully you enjoy the rest of the presentations.
Thank you, Vanessa.
Mhm.
Our next presentation will be by Victoria Eaton.
Hello, everyone.
Good morning.
Can everyone see the presentation?
Yes.
Good morning or good afternoon. My name is Victoria Eaton. I'm a rising sophomore computer science major. And this summer I did a literature review project with Dr. Jason DeBryan, who's at Morehouse School of Medicine, looking at the paper titled Ubiquitin Ligase SIA2 Regulates Reverb Alpha Degradation and the Mammalian Circadian Clock. So just to give a brief, broad overview, it's known that the circadian clock proteins are degraded by the proteasome. And so the lab looked at the reverb alpha, which is also called NR1D1, which is a crucial protein for the proper function of the mammalian circadian clock. And so the research question was, what controls reverb alpha circadian degradation? And the hypothesis was that it was an E3 ligase or E3 ligases. And so they used a cell-based screening approach to identify those E3 ligases, and found that SIA2 and SBSB4 were both E3 ligases responsible for the degradation of reverb alpha. And just to go more in depth into what's happening, so the process by which proteins are degraded by the proteasome first starts out with an E1 ligase, and that E1 ligase attaches to a ubiquitin molecule using ATP. And then once that is done, then the E1 ligase transfers that ubiquitin onto an E2 ligase, and that E2 ligase then attaches to an E3 ligase. And so then the E3 ligase has the E2 ligase with the ubiquitin on one end, and on the other end it has a protein. In this case, that protein was reverb alpha. And so then after that, the E3 ligase attaches the ubiquitin onto that protein, reverb alpha, and it becomes polyubiquinated. And then that protein is then transported to the proteasome where it's broken down. And so the lab looked at reverb alpha, which is a nuclear hormone receptor, and its protein and mRNA levels were measured during the experiments. And the goal was to find the E3 ligase responsible for the degradation of any given protein of interest using a functional screening approach. And the results, of course, found that SIA2 and SBSB4 mediated reverb alpha turnover. And so the cell-based functional screening approach looked like this. Here in figure A, you can see this is to represent an empty well. And so all those gray dots are empty wells. And so they took Cry1, which is another circadian clock protein. They've implanted Cry1 into each of those wells. And then they took a library of over 700 different E3 ligases, and they implanted it into that well with Cry1 to see if that E3 ligase would degrade Cry1. And so after 2 days, the production of new protein was stopped. And then they went and looked at where was Cry1 no longer present. And so the black dots are the wells where Cry1 was no longer present, and the yellow parts are where Cry1 was still present. And so the black dots, you can infer that that means that the E3 ligase that was put into that well was an E3 ligase for Cry1 since it degraded Cry1. And so you can see in figure B, RNF128 and FBXL3 were both E3 ligases that degraded Cry1. And so once this method was found to be validated and found to be effective, they then did the same thing for reverb alpha and found that SIA2 and SBSB4 were both E3 ligases that degraded reverb alpha. And so there are three factors that make an E3 ligase. One, it has to interact with the protein, it has to ubiquitinate the protein, and it requires ubiquitin ligase activity. And so the lab did experiments to prove that basically each of these were, that the experiment checked off each of these boxes essentially. And so this figure right here, you can see it used SIA2 siRNAs. And what these siRNAs do is they essentially silence SIA2, meaning that the more siRNAs that are present, the less SIA2 is present. And so this experiment basically shows how the less SIA2 you have, the longer the circadian rhythms are. And you can see, especially around day 4, 5, and 6, is where the circadian period lengthened. And so over here in figure D, the same experiment was done. Basically, it's showing that SIA2 increased the period length, SBSB4 also increased the period length, and then both were depleted, it increased the period length the most. And so you can see when SIA2 was depleted, the period lengthened around 2 hours. When SBSB4 was depleted, it lengthened around an hour. And then when both were depleted, it lengthened the most, over 2 hours. And so both of these experiments basically suggest that SIA2 and SBSB4 depletion delayed the circadian rhythms. This method was used because previous methods had gaps in its ability to determine E3 ligases for specific proteins. Previous methods were more useful for identifying the role of the E3 ligases and not the E3 ligases themselves. And so this method does not require stable protein interactions, making it fast, specific, and able to identify E3 ligases for any given protein. This method is important because general screening approaches in the past have lagged, and with this new method, it can be applied to circadian clock related diseases like insomnia to see more specifically what is going on. And so the next step for this research, since SBSB4 was identified as an E3 ligase for reverb alpha, the next step would be to conduct further research to identify the role of SBSB4 in circadian periodicity and reverb alpha regulation. Here's my reference slide. I only used that one paper. And I would like to thank Dr. Jason O'Brien at Morehouse School of Medicine. He's in the pharmacology and toxicology wing, and he was very crucial in my understanding of this research. If it wasn't for him, I don't think I would have gotten a great of experience as I did this summer. And I'd also like to thank the RISE program for giving me this opportunity to really get experience reading scientific papers, which is what I'll be doing in grad school. If there's any questions, I can answer them right now.
I have a question. So did you play a role in conducting this research since it's like from the lab that you work in now, or was it like given to you since we're virtual?
Yes, so this paper was actually done and it was published in 2015, and I didn't have a role in doing any of the research or doing any of the experiments, but my role in the lab now does relate to this. It does relate to this research, but I do more of a computer science aspect since I'm a computer science major. Are there any other questions?
Hi, yes, I have a question. Hi. I would first like to say great talk, and can you talk a little bit more about the computer science aspects that you worked on?
Right, so when I went into the lab last semester, I was looking at how the research kind of, it did go on further, it developed more. So I was looking at specific transcriptional factors, and my role was to identify what those transcriptional factors were using computer science software. So I was working on downloading the software called Homer, and it would be used to identify those transcriptional factors, but I didn't get much into that since the whole COVID thing happened.
Thank you.
Mm-hmm. All right, well, if there's not any more questions, I'd like to say thank you for your time and enjoy the rest of the presentations.
Thank you, Victoria. Our next presenter is Jacqueline Willis.
I'm gonna share my screen now. Okay, can everyone hear me?
Yes.
Okay, just wanna make sure. Okay, hello everyone. My name is Jacqueline Willis, and this summer I was a SHIRT participant at Harvard Medical School. I worked in the lab of Dr. Muthuswamy under the mentorship of Dr. Wong. My research is titled Exosome Associated Proteins as a Biomarker for Pancreatic Cancer Diagnosis. So today's presentation, I'm going to go through an introduction and a purpose, go through my background information, my hypothesis, methods, results, conclusion, and talk a little bit about my summer experience. So hello, as I said, my name is Jacqueline Willis. I'm a senior biochemistry major. This upcoming fall will be my last semester. I have been a RISE scholar. This is my second year now. At my home institution, Spelman, I am a researcher in Dr. Hibbert's lab. My career goals are to become a physician scientist, specifically to pursue my MD, PhD, and my after graduation plans are to be a researcher at the NIH as a UGSP scholar. So the purpose of today's research is to identify an extracellular vesicle associated biomarker list for diagnosis of pancreatic cancer. So secreted vesicles are small membrane enclosed sacs that store and transport substances from cell to cell. Once the vesicle leaves its original cell, it can fuse and dock with the membrane of another to release its contents, which is what is being shown here in this image, the two different pathways. So the secretory vesicle can either fuse with the membrane and release its contents, known as exocytosis, or it can form and the vesicle can be indulged within the cell, which is known as endocytosis. Exosomes are a type of secreted vesicles, and exosomes are small extracellular vesicles with a multivesicular endosome origin. And so here in this image, as we see, so once the exosome fuses with the membrane of its cell, endocytosis happens, and it causes the exosome to fold inward into itself into a multivesicular body. And there's different pathways that this multivesicular body can go through. It can either form with the lysosome to be degraded, or it can continue exocytosis and be secreted as an exosome, which is what we see here in this image around letter C. Exosomes contains many different contents. So here we see on the surface of exosomes are their proteins and lipids, and within exosomes are nucleic acids and different metabolites. So here, this image is just showing a healthy pancreas versus a malignant pancreatic tumor. And so recent studies have shown that in healthy patients, the level of exosomes produced are about 2,000 trillion, but in cancer patients, that number is said to be doubled. And so the problem here that we're trying to focus on is there are little to no reliable methods for detecting early stages of pancreatic cancer, because of the location of the cancer. Is inside the body and it's not easily detectable. And so here, this image is just showing different roles that exosomes play in cancer. And so exosomes can aid in angiogenesis and metastasis of tumors, evade the immune response, cause cancer-associated fibroblasts, and also has a role in epithelial to mesenchymal transitions, which also adds in metastasis of cancer. And here are just a few cancer biomarkers that can be used to detect cancer or used as therapeutic targets. So like proteins, nucleic acids, metabolites, and different organic compounds. And so the hypothesis that we came up with is that proteins found in exosomes are overexpressed in the blood of pancreatic cancer patients, as well as these proteins can be used as a diagnostic metric for early detection of pancreatic cancer. And so the methods, as you know, this summer, the research was virtual. So the portion that was done, to give a little bit of background about the portion that was done in the lab. So my lab works on 3D models of tumors using organoids. So a pancreatic tumor organoid was created to collect purified exosome samples. And then proteomic techniques was used to create a proteomic profile of purified proteins from the sample. And so I received that list from my mentor, Dr. Wong, and then virtually I used a collection of C-Viral Portal Studies, which is a website that is a collection of different cancer studies in one place. Um, and I used it to identify overexpressed proteins from pancreatic tumor organoid samples. Next, I used another collection of C-Viral Portal studies to validate the list of proteins found to be overexpressed in the pancreatic cancer types. And once all of this information was created, I collected the information and created an official data sheet. And so here's a result of the literature search that I did on one of the genes, which is MUC1. MUC1 acts as an adhesion and anti-adhesion protein in addition to its role as a protective layer on epithelial cells. This gene or protein is predominantly localized in the extracellular regions, the nucleus, the plasma membrane, and the Golgi apparatus. And the role that MUC1 plays in cancer is often overexpressed in AIDS and metastatic cancers. And so because it acts as a driving force in the changes of the cytoskeleton and adhesion capacity of the transformed cell, which can aid in metastasis. And so MUC1 is often used as a diagnostic marker for metastatic progression. And so here what this graph is that I created is it's showing the Z-scores of the MUC1 mRNA expression average. And what I found interesting about this data is that the liver, MUC1 in the liver was downregulated. Well, this research is more so focusing on pancreatic cancer, but I thought it would be interesting to look a little bit more into why the liver MUC1 gene protein expression was lower than the others. And here, this is an image of looking at the overall survival comparison of altered versus the unaltered group. So the way C-Bio Portal works is I was able to go through and click a bunch of different studies, and I clicked studies for liver cancer, lung cancer, prostate cancer, pancreatic cancer, and breast cancer. And so what I did was look at the protein expression in these different cancers, as well as looking at the survival rate. And so the p-value in this data was said to be significant. And as we can see here from the graph in the lower right corner, that out of the 211 patients of all 5 studies, 59 of those patients died. 59 of those patients died from having this protein mutation. And so our conclusions and future directions for this research is, so these protein biomarkers will be useful in diagnosing the early stages of pancreatic cancer. I'm also looking for successful antibodies that may lead to these proteins becoming successful therapeutic targets, as well as the lab would like to develop an assay for one of these proteins for the early detection of pancreatic cancers. And I didn't mention this, but it's not just, I only presented one of the proteins, but I actually have a list of 18 that I'm going through. I'm grateful to my PI, Dr. Muthuswamy, and my mentor, Dr. Wong, for their guidance. My work was supported by the RISE grant, so I'm very grateful for that. And I would also like to give a thank you to Dr. Karina Gonzalez, Dr. Sheila Thomas, and the Harvard SHIRT program for the opportunity to conduct research this summer. Just to talk a little bit about my experience this summer, I was blessed enough to be able to participate in 2 internships, one as a SHIRT participant at Harvard Medical School, and one as a cancer participant at the University of Colorado. Both of these, although virtual, both of these experiences gave me the soft skills and helped me hone in on my skills for literature searches and just different skills that I felt like I needed to work on before I entered graduate school. I'm grateful for the SHIRT program because they have us working on an NSF grant before we enter and apply for graduate school. We're also taking the art course and have had many opportunities to network. So I'm very grateful for that. And as well as the Colorado Medical School internship, we have had weekly cancer modules learning about different cancers. So that has definitely aided in my different interests of the science field. And so I'm very grateful for that as well. But that is all. If you have any questions, please ask them. Thank you.
Thank you, Jackie. Any questions?
Hi, Jackie. How are you doing? I have a couple of questions.
Hello, Dr. Jackson.
You sounded afraid when I said that.
I am a little.
But were you able to create any of the organoids? I wasn't sure because I'm trying to create some prostate cancer organoids.
No, I wasn't. I'm not even sure how that process works. I haven't been like told the methods or anything. I just was told that the organoid sample was created.
Okay, fantastic. And the other thing that MUC1 or Mucin1, there's a big team at Yerkes doing that work. So if you want to, hold on just a moment. Sorry about that, I have teens. So do you know whether or not if you want to, I'm just saying if you want to continue this work, there's a big team at Yerkes that's working on the MUC1 genes. I want to continue working with them. I'm just giving you that as a heads up, okay?
Okay, yes, thank you. And it's at the school is Yerkes University?
It's with Emory, Yerkes.
Okay, thank you.
There. So they're doing some work with MUC1 in immunology and some in cancer, okay?
Okay.
So, and congratulations. I'm sorry, everyone, I have to leave out. Congratulations to the chemistry, biochemistry majors, my Link scholars. I see you guys. I have to go to another meeting now, but congratulations, this was great.
Thank you, Dr. Jackson.
Are there any other questions? Okay, I would like to thank everyone for listening to my research presentation and hope you guys enjoy the rest of your evening. Thank you.
Thank you, Jackie. Our next presenter is Noah Odell.
All right, good morning, everyone. My name is Noah Odell. This summer, I had the privilege of being in the virtual summer research program at the Indiana University Simon Comprehensive Cancer Center. My mentor was Dr. Timothy Corson. He's the Associate Professor of Ophthalmology at the IU School of Medicine, and my summer project was on the circadian effects of choroidal neovascularization in a mouse model. So a little background information about me. I'm a rising sophomore at Spelman College. I'm a biology major on the pre-med track. I'm a part of the Health Careers Club and the NAACP Health Committee. When I'm older, I want to be an OBGYN or a geneticist. I was a part of the summer research program at IU last summer as well, where I was also in the Corson lab, and I'm part of the RISE program at Spelman. And after graduating, I want to pursue an MD PhD. So, small disclaimer, the views and findings that I'm about to present are entirely my own and do not reflect my mentor, the research group, or any affiliated institution. So my summer project was kind of two parts. The first part was an annotated bibliography where I looked at articles related to retinoblastoma and angiogenesis. And the second part, which will be the focus of my presentation, was my circadian effects on neovascularization project. So one of the things that the Corson lab focuses on is retinoblastoma, which is a pediatric eye cancer that typically develops before the age of 5. There's about 200 to 300 cases per year in the United States. It can affect one or both eyes. You can either inherit the gene for it, or if it appears randomly, we call that sporadic. There is no cure for retinoblastoma, though there are multiple treatment options, which include chemotherapy, freezing or burning a small part of the tumor, or enucleation, which is just removing the eye altogether. So in the corner here, you can see a picture of a little boy who has the retinoblastoma tumor in both of his eyes, and that's like the white calcified part. So 2 words you're going to hear me say a lot are neovascularization and angiogenesis. So the difference between the two is that angiogenesis is supposed to happen within the body. It's just the development of new blood vessels. But neovascularization is bad angiogenesis. It's the development of blood vessels where the tissue has been impaired by trauma or disease. And so it's a feature of several eye diseases like proliferative diabetic retinopathy, neovascular age-related macular degeneration, and also retinopathy of prematurity as well. It's sort of a hallmark of cancer. It occurs in all cancers, not just eye cancer. And so for it to happen, these new blood vessels need to recruit oxygen and nutrients from the body, which helps it to spread. So one of the types of neovascularization that I focused on was choroidal neovascularization, also known as CNV, which is the growth of new blood vessels that originate from the choroid through a break in the Brooks membrane of the eye. So as you can see in the right-hand corner, I have a picture of an eye, and the choroid is right below the retina. It supports the retina and also gives it its blood supply as well. So what that has to do with retinoblastoma is that the blood vessels are needed for tumor growth. And so neovascularization can take advantage of the retinoblastoma tumor and help spread the cancer throughout the eye. So this other picture that I have down here at the bottom is a picture of a live mouse eye that has new blood vessels growing in and on it. So circadian rhythm, we all know that we have a sort of internal clock inside of us that repeats roughly every 24 hours. And as the picture shows, it's related to our sleep and wake cycles, body temperature, blood pressure, and hormone secretion. So disruption of this clock can create environments for cancer to spread because some critical genes, like genes for cell division or cell proliferation, are regulated by circadian rhythm. And so there have been studies that have shown that disrupting the clock has also been correlated with a heightened risk of diseases and conditions like obesity, depression, and cancer. So we wanted to know if there are circadian effects on neovascularization. So my question was, is neovascularization subject to circadian control? And to look at that further, we used an LCNV model, which stands for laser-induced choroidal neovascularization, where they aim a laser at the Brooks membrane in the choroid of the eye, and they rupture it to induce angiogenesis. And so the LCMV model can be applied to many aspects of neovascular age-related macular degeneration. And so you can look at things like molecular mechanisms, the effect of genetic manipulation and drug treatment effects. And so those are the images that I have over here in the right-hand corner. And so the laser made these 4 lesions on each eye, as you can see in these 2 images. And then we also used OCT imaging for other pictures and to get the volumes of the lesions. So over here on the left, we have an OCT image. When OCT is using light waves to take cross-section pictures of the retina, that way you can see the retina's distinctive layers, and it allows you to map and measure its thickness. And then with these measurements, it can help with diagnosis and provide treatments for glaucoma and then diseases of the retina like retinoblastoma. So this red circle would be the choroidal neovascularization happening in the eye. And so what I did was I took the volume of each of the lesions in these images and I put them against the time that the laser treatment was done. And so you can see this first one was done at 10:41 a.m. and this second one here was done at 1:56 p.m. So we wanted to see if the volumes changed based on what time the laser treatment was done. So the results that I had was there was only a slight decrease over a 4-hour period. I'd like to say that my analysis that I did was a secondary analysis. This project was not originally done to maximize circadian effects. So there is only a small window of time for these treatments. And so this R just shows that it's only a slight decrease. And then this p-value here shows that there was no significant change at all. So discussions and conclusions. The conclusion I came to was the data was not sufficient enough to determine that neovascularization was under circadian control. But like I said, my analysis was purely a secondary analysis. So if someone was to redo the experiment to maximize circadian effects, they could create a greater time gap between the images. So instead of just doing a 4-hour time span, they could do it over a full 24 hours. But the fact that there wasn't really a significant change is kind of a good thing, because when you think of future experiments and future treatments, the time of day that you do the treatment or do the experiment shouldn't affect your results that great. So how this summer experience helped my path to a PhD, it gave me another chance at having research experience. It enhanced my reading and writing skills. I was able to talk to a wide variety of people with MDs and PhDs, and they were able to give me career advice. I was able to establish relationships with them and also gain a few mentors along the way. And I was able to be exposed to different fields throughout the various seminars that I attended throughout the summer. These are my references. And so I'd like to thank Dr. Corson, Kamakshi Shisla, and the entire Corson Lab, and also the Spelman RISE program that was funded by the RISE grant. And then I'd also like to thank the IU Comprehensive Cancer Center for allowing me to participate in this research experience for another summer. Thank you for listening to my presentation. I will now take questions.
I have a question. So how many mice did you end up analyzing, and did you evaluate the volume in both of their eyes or just one?
I was given the data for 32 mice, and then I evaluated both the left and right eyes. The pictures that I had in the PowerPoint were just the left eyes, but I matched the volumes of left and right.
Other questions? All right, if there are no other questions, thank you so much for your presentation, Noah. And our next presenter is Jaya Fowler.
All right, everyone can hear me?
Yes.
Okay. Hi, my name is Jaya Fowler, and this summer I worked with Dr. Davida Camp on fragment-based drug design of the ISD heme uptake system of Staphylococcus aureus bacteria. So for some background about me, I am a rising sophomore chemistry major at Spelman College, and I'm currently presenting from Roswell, Georgia. My future research interest and the research focus of our lab is in medicinal chemistry. And as aforementioned, my research mentor was Dr. Davida Camp, who is of the chemistry department at Spelman College. And for some background about our project, Staphylococcus aureus is a gram-positive bacteria that is frequently found in the upper respiratory tract and/or on the skin. However, Staph aureus is particularly dangerous because it has the ability to invade the human bloodstream. When this happens, the bacteria can cause severe illness, including fatality. Iron is the primary nutrient of the ISD heme uptake system, and it is responsible for the growth and spread of the bacteria. Staph aureus has not only developed drug-resistant strains, but it also disproportionately affects minority communities. So our lab is focusing on this particular system of proteins to hopefully come up with an effective treatment for the bacteria. So as aforementioned, Staph aureus contains the ISD heme uptake system, which is a system of proteins that assist in the rapid spread of the bacteria. ISDBN2's primary function is to abstract heme from hemoproteins, which are essential to the growth of Staph aureus. Our project focuses on designing a small molecule inhibitor that prevents the protein from attracting heme and helps to stop the growth and spread of Staph aureus. We are using a fragment-based drug design approach to analyze interactions between natural products and critical residues found at the binding site to synthesize our small molecule inhibitor. My project this summer focused on analyzing the inhibition constant and binding affinities of these natural compounds to better understand the molecular structure of our binding site and which functional groups of the compounds contain characteristics of fragments we want to dock in the future. So for our research method, so this diagram shows two different approaches that we can take to designing our small molecule inhibitor. To the left of this diagram is a high throughput screening approach. And with this particular method, you dock large compounds into the drug target or binding site, and you analyze the different interactions or hits that the compound has with the protein. And then you can develop a biochemical assay on these interactions. Some issues that were associated with this particular approach that drove us away from using this one is that it only sparsely samples the chemical diversity space of our protein. you have a likelihood for having non-drug-like parameters, and then you also can avoid, or you also can encounter more unavoidable interactions between your protein and your compound. So to the right of this diagram is a fragment-based drug design approach, which is the approach that we're taking to design our small molecule inhibitor. And with this particular approach, you dock smaller fragments into the drug target, and then you link these fragments together to synthesize your compound. With this particular approach, it's used with elements that have a strong structural characterization. It's more time efficient, and then you also can avoid unavoidable interactions between your protein and your compound. So just to repeat, I analyze which functional groups of the natural products that I docked into our protein better interact with our binding site to give insight on which fragments we can dock in the future. So this is AutoDock Tools. This is a program that I primarily use to dock the natural products or the compounds into our protein. And so this basically is a program that analyzes different positions in which the compound can be docked into the binding site of the protein, and it provides computational data on the binding affinity that the compound has to the protein. And this image is actually protein chain A of the protein that we use out of the ISD heme uptake system. And then in the blue box to the right of the molecule is where I docked one of the compounds. This next program is called PyMol, and this is a molecular visualization system, and I use this program to get a closer look structurally at the interactions between our compound and our protein that was docked into the binding site. So for our data and analysis, our data retrieval process went as followed. I first docked 3-RTL, which is our protein, into AutoDock, and I prepared it by deleting the water molecules, adding polar hydrogens, and then adding Coleman charges. And this basically prepares the protein to accept a small molecule or a compound, and it also stabilizes the protein. I prepared the ligand in the same way, and then I docked the ligand or the compound into the protein, and I docked 10 different positions through AutoDock, and they were ranked by their binding affinities. So these are the 6 different ligands that I chose from the zinc database to dock into our protein. I chose these particular ligands because they follow Lipinski's rule of 5, which means that they have no more than 5 hydrogen bond donors and 10 hydrogen bond acceptors, and this ensures drug likeness of the compound. So this is actually output data from the first ligand that I docked, zinc 7, into our protein, and I organized it by the estimated free energy of binding and the estimated inhibition constant. So out of the 10 different poses, it provided me with each position or poses free energy of binding and inhibition constant. When analyzing the output data of the compounds, we want to look for the positions that have a small inhibition constant. The inhibition constant tells us how much of the compound is required to inhibit the original function of the protein. We want to use the smallest amount possible to ensure drug likeness of the compound, yes. So essentially, the less of the compound that the human has to ingest when we synthesize a molecule to become a drug, the better. The free energy of binding tells us how well the compound binds to the protein. And so the closer to 0 this number is, the higher the binding affinity the compound has to the protein, and that's also a good thing that we want to look for. However, with our particular experiment and our particular aim, we want to put the inhibition constant, or the inhibition constant has significance over the free energy of binding for the data that I'm looking at this summer. So I primarily looked at the inhibition constant when analyzing which position of the ligand would be best in the binding site. So for zinc 7, docking pose 9 is the best out of the 10 docking poses because it provided us with the lowest inhibition constant of 360.25 micromolar. So this chart is a summary of the data that of the 6 different compounds that I docked and their best docking position based on their estimated inhibition constant. I provided the estimated free energy of binding just to compare and get more information for future analysis. But based on this data, Zinc 14 contains functional groups that we want to look for in fragments that we dock in the future because it has the lowest estimated inhibition constant out of the 6 compounds of 165.21 nanomolar. So essentially that means only a little bit of this compound is required to inhibit the function of the protein. So for the discussion, the overall goal for my summer research was to analyze the binding interactions between natural products and the heme binding site of the ISD heme uptake system of Staph aureus. And in conclusion, I was able to determine which natural products out of the 6 I analyzed interacted best with our binding site, and this will show us which functional groups, well, with further analysis, which functional groups have better interactions with our critical residues, and we can use this information to apply to which fragments we can dock in the future to synthesize our small molecule inhibitor. So for future steps, using this information in further analysis, we can actually choose and dock fragments, smaller fragments into our binding site, and then we can link these fragments or synthesize our molecule, and then we will do biological assessments on our synthesized compound with other compounds to see different interactions that it has. So for my personal experience this summer, I included this quote by Thomas Edison that says, "I have not failed, I've just found 1,000 ways it won't work," because Dr. Camp and I wouldn't say we went through a lot of trial and error, but we did a lot of work after or transitioning to fully virtual studies because, you know, I used to be working in the lab before we had to do quarantine. And so transitioning from doing primarily experimental studies to computational data, I learned a lot about the significance and importance of understanding your computational studies and as well as using this information to be put at a better position to do experimental studies in the future. So this experience has taught me a lot about how to, I guess, go through a proper and efficient and accurate research method and research study. And so I'm excited to apply the information that I learned to further research in the future that I continue to do with Dr. Camp and that I continue to do in graduate school throughout the rest of my career. And so I would like to acknowledge the National Institute of Health, the Spelman College RISE program, Dr. Blanks and Ms. Bird, Dr. Brennan, and Dr. Camp for making this research possible. These are my references, and that concludes my presentation. I will accept any questions.
I just want to say very good job in your presentation, Jaya. And my question was, what kind of further analysis do you plan on doing with that data that you collected this summer?
So I mentioned this in the earlier in the presentation. Using this information out of the 6 zinc or natural products that I docked into our protein, I can analyze which functional groups out of the structurization of these compounds were interacted best with our binding site, and I can choose fragments using that information choose fragments that best interact with our binding site using the information that I found from docking these natural compounds into our protein.
Thank you.
Thank you. So if that's all the questions, thank you for listening to my presentation.
Thank you, Jaya. Our next presenter is Aaliyah Brown.
Okay, good afternoon, everyone. Can everybody hear me?
Yes.
Okay. My name is Aaliyah Brown. I'm a sophomore psychology major from Atlanta, Georgia, and the title of my presentation is Art and Perspective, Enhancing the Aesthetic Experience with Contextual Information. So just go ahead and get started here. Our research seeks to find a better understanding to how information and art intersect to provide meaning. We're trying to do that by answering the question, to what extent does contextual information impact or enhance the aesthetic experience in art museums for African American college students? Why is this research relevant? In society today, the educational system often forces students to a traditional style of learning that can often be non-conducive and produce a lot of self-esteem issues that students and young adults have to overcome. Additionally, many teens and young adults struggle with various psychological disorders, and while we already know that art therapy can be helpful for them, it has only been studied in the lab context or the therapeutic context. And through a literature review, I learned that museum contexts yield different results than the lab or therapy, and they're heavily understudied as the field of study of art and aesthetics is relatively new. Further introduction and hypothesis, the literature review. So this summer I conducted a literature review, and I learned that aesthetic experience in art museums is understudied, creating a need to move out of, I blocked my presentation. I need to move out of the lab to examine the relationship and response to art in a more natural environment like an art museum. Studies have been done comparing the impact of descriptive information and contextual information in art museums for time spent viewing art and reading labels. Viewing art and exhibits in the museum context can provide a fresh, non-traditional way of learning. Art and aesthetic experience can be directly linked to well-being, and viewing art in a museum can be directly related to physical healing and pain relief. So how is our study authentic or novel? We are examining the impact that contextual information has on aesthetic experience when viewing art and using that research for future studies in art museums. So further introduction and hypothesis, the gap in knowledge, our research shows the gap in knowledge about the extent to which contextual information impacts the aesthetic experience. Research on contextual information in art museums has been done with adults and abstract art. However, our study, we will examine the response in African American college students with contemporary mixed media. We propose to examine college students' responses to Deborah Roberts' art exhibit, The Evolution of Mimi. Deborah Roberts' The Evolution of Mimi is a series of mixed media collages that depicts the struggle of African American girls and the things that they have to overcome regarding beauty standards and stereotypes in America. We hypothesize that adding information to emotionally and racially charged artworks will enhance the viewer's experience and impact their interpretation, opinion, and appreciation for the art. So on the right here, we have an example of one of Deborah Roberts' art collages. And for our methods, we'll be using an experimental design to create two websites, one with context and one without, to measure the impact the aesthetic information has on their aesthetic experience, the contextual information has on their aesthetic experience using the Survey for Assessment of Aesthetic Perception, or the SAP scale. Participants will be randomly assigned to either one of the sites. And then about the SAP scale for psychometric data validity and reliability. Psychometric data collected on the SAP scale indicate satisfactory, reliability, and validity. Construct validity was established with principal components analysis, yielding 3 factors, with the first factor explaining 39.2% of the variance as a result of Varimax rotation. Internal consistency estimates ranges from 0.69 to 0.83. And inter-rater reliability. As measured by Cronbach's alpha, we were also in the accepted range for reliability. We have chosen to use Deborah Roberts' The Evolution of Mimi in her art because the art is not really abstract, but it is still open to interpretation. And it's also racially charged, like I mentioned earlier, and emotionally charged, hopefully. A couple of our limitations: we're only using one artist, only one type of art. We have a limited participant pool, and participants are viewing art online. And because we're using an experimental design, we have a lot of internal validity and not a lot of external validity. However, we hope to increase external validity in our future studies. So I wanted to take you guys to the site that I've been working on Can everybody see it?
Yes.
Okay. So the title of the site is Art and Perspective, Examining the Aesthetic Experience, a pilot study on aesthetics. At the beginning of the presentation, beginning of the site, there's just a couple of demographic questions to see. We're going to see if those influence the data in any way. Then next, there's a brief paragraph describing Deborah Roberts and her art exhibit and some of her works. Then right under that is 8 images that Roberts have created, and under each image is a quote from Roberts, either about the exhibit as a whole or about the individual piece. So participants will look at each image and read the quotes. And then under that, there is the questions that come from the scale that I mentioned earlier. There's about 15 questions or so, and the options range from totally agree to totally disagree. Back to my presentation. Our proposed analysis, we plan to run a one-way ANOVA to test for differences between the 2 groups, those that respond to the context website versus those who respond to the website without context. And we plan to see if there are correlations between the responses and demographic data such as choices major or minor, year of matriculation, and or previous experience with art in a series of Pearson's bivariate correlations. If the results from the study reveal significant differences, we will accept our hypothesis. If they don't, we will reject our hypothesis and do further study. For implications, hopefully our pilot study will give us some foundational insight on college students and their responses to art and information and the way they intersect. With this insight, we will be able to better create an app that enhances learning and hopefully contributes to overall more impactful experience. As you know, being in the physical museum produces different outcomes than experiencing art in other ways. So it is beneficial and important that we eventually do the research in the right context. Because of this, we are planning on doing an in-person study with pencil and paper survey to measure the aesthetic experience of the museum and then compare it to that of this study. After that, finally, with all this information, we plan to create our own app for AUC museums and exhibits. The app will have survey questions about its effectiveness, as well as questions about other demographic information to see if that influences our results and add an extra component of study. These are my selected references. And my acknowledgements, I would like to give a big thank you to my mentor, Dr. Andrews from Morehouse College. It's been just me and her this summer, so I've gotten a lot of attention. I've got to ask all the questions that I needed, and I got to learn so much from her. I would also like to thank Dr. Blankson, Dr. Brennan, and Miss Bird in the RISE program. And then my RISE 2020 summer experience, my highlights from this summer. I've done a lot this summer. I've created a literature review, I've done an IRB, I've developed a website, I've done this research proposal, and I've also had a lot of helpful and informative modules that taught some of the intangible skills of being in a lab and working with a mentor. And this all prepared me for a PhD program in the future because it's taught me how to work with a mentor and develop a good relationship with one. And then I also learned a lot about the process and all the work that has to be done before you conduct an actual study and why all that background work is really important. And then this summer has also taught me just how to be adaptable and flexible and be determined to get work done. Originally, me and Dr. Andrews planned to do work in person, and I didn't think that I would have to develop a website, but I have, and it's been a very cool experience. So thank you. Any questions?
Great job, Aaliyah. I have a quick question. What was the biggest challenge that you had to overcome while doing this research?
The biggest challenge that I've overcame this summer, I think, was actually just, getting comfortable with a mentor and getting comfortable in the lab, or not in the lab, but, getting comfortable talking to the mentor and doing the literature review, just like getting the timing down, how to read a paper effectively and go through the figures effectively and stuff like that.
Thank you.
Thank you.
Hi, Aaliyah. I have a question for you. So do you plan to continue conducting this study in the fall?
Yes, so the IRB we made this summer will hopefully be submitted soon, and then we'll get approval, and then hopefully we'll be able to get some participants to actually work on or go through the 2 websites that we made this summer so we can collect data and continue our research on time.
Thank you.
So any more questions? If not, thank you guys. Enjoy the rest of the presentation.
Thank you, Aaliyah. Our next presenter is Ayana Forte.
Good morning or afternoon, everyone.
Good afternoon.
Hello, everyone. My name is Ayana Forte, and this summer I had the pleasure and opportunity of working under the mentorship of Dr. Kimberly Williams with the NIH program virtually this summer. The title of my presentation is HIV and Neuroinflammation. A short introduction to myself is I'm a junior at Spelman College, majoring in psychology, minoring in Spanish. I'm from Chicago, Illinois. I'm going on my 2nd year with the RISE program. The beginnings of my research project started in the Shudo lab at the University of Pennsylvania and continued on to the KSW lab at Spelman College. I'm also an advocate for mental health. The focus of my research this summer was on HIV, also known as human immunodeficiency virus, which is a sexually transmitted disease that can lead to AIDS. HIV has infected approximately 30 million people worldwide currently, and a majority of these cases are located in Southern Africa. If we look to this diagram to the right, we can see the highest prevalence in the red color is seen in regions of Southern Africa. Although CART medication has shown to be successful in treating HIV and prolonging the lifespan of these patients often experience many comorbidities from the virus, such as immune disorder, PCP, and HIV-associated neurocognitive disorders, otherwise known as HAND. Even after the introduction of CART medication, 40 to 50% of HIV patients still suffer from some form of cognitive impairment, broadly known as HAND. HAND is a spectrum of disorders ranging in severity from the most severe form, which is HIV-associated dementia, to mild neurocognitive disorders, MND, and the least severe being asymptomatic neurocognitive impairment, or ANI. Mild neurocognitive Prior to CAR treatment, 50% of patients suffered from HAND, and a third of the patients had patients, the most severe form. However, after CAR treatment, we see that less patients experience HAD, but still 50% of patients are suffering from some form of the disorder, causing cognitive problems. And this age is also known to or expected to increase with aging. Some of the effects of HAND are changes in one's quality of life, which these changes can be exacerbated when patients have experienced previous traumas. A study that I found this summer told a story about a woman who was admitted to the hospital for catatonic behavior and fluctuations in her consciousness after she had been living years of a healthy life with HIV virus. When she was admitted to the hospital, doctors conducted a spinal tap and found that her plasma viral load was 60 copies per milliliter, which is a healthy range for HIV positive patients. But in her CSF, her viral load was 45,600 copies per milliliter. This was later found to be known as HIV viral escape syndrome. HIV viral escape syndrome occurs when the virus escapes to the brain by crossing the blood-brain barrier. However, the combined antiretroviral therapies cannot cross the BBB, which leads to neuroinflammation. Neuroinflammation is the introduction of external stimuli, injury, or infection into the CNS, which can lead to inflammation, neuroinflammation. Some of the key hallmarks of neuroinflammation, as seen in this diagram, are immune cell infiltration into the CNS, which activates the astroglial and microglial cells, which then leads to the release of cytokines, chemokines, and other neurotoxins, which leads to neuroinflammation. Previous studies in our lab allowed us to form an in vitro model of neuroinflammation and HIV infection, where we were able to infect human macrophages with HIV virus, collect their supernatants, which will be holding the neurotoxins that they're emitting, and put this media onto rat cortical neurons to assess neuronal damage. In this image to the left, we see on the left side, we have untreated neurons, which can be seen to be healthy and abundant by the abundance of green color from the MAP2 staining of neurons. In comparison to the right, we have neurons that have been exposed to media from HIV-infected macrophages. And we can see that there's a significant decrease in the green color, meaning that there's a significant decrease in our neuronal count, which we can see that there was damage to the neurons in this image. This model can also be used in other diseases associated with neuroinflammation, such as Alzheimer's disease, Parkinson's disease, and PTSD. Not many studies focus on how other disorders can exacerbate neuroinflammation in HIV patients, and our lab is interested in how PTSD influences HIV-associated neuroinflammation. Another study that I found this previous summer studied the association between PTSD and inflammation, as well as immune activation in HIV-positive, virally suppressed individuals. The study found that there was a high prevalence of PTSD amongst these participants. About half of them reported trauma exposure, and about half developed PTSD. These results suggested that PTSD may be associated with immune dysregulation. And among HIV-infected individuals, it has been found that PTSD is associated with greater or faster morbidity. In fact, incidence of stress-related disorders such as PTSD is elevated amongst those living with HIV versus those living without HIV. The diagnosis of HIV in itself is often characterized as a form of a traumatic event. Given the connections between HIV, PTSD, and neuroinflammation, some of the future directions for my project in the KSW lab is to elucidate the role of neuroinflammation in donors experiencing these disorders. And to do so, we will utilize our in vitro model system to understand how psychosocial, genetic, immunological, and neurotransmitter systems can interact to help target new preventative and symptomatic therapies. My research this summer helped me to move another step forward towards my PhD by helping me to strengthen my critical analysis skills, become more aware of the data about HIV, neuroinflammation, PTSD, and more. I became better and more efficient at reading articles and studies, And this summer also allowed us to build more narrative onto the previous findings in our lab. Some of the people I would like to acknowledge are my research mentor, Dr. Kimberly Williams, for being a great mentor to me and a great person in general. I'd also like to acknowledge the RISE program and NIH and the members of the RISE program that keep the program running so smoothly and for giving us this opportunity. These are my references. Thank you so much for your time. I'd now like to open the floor to any questions or comments.
Thank you, Ayana. Are there any questions?
I have a question.
Yeah.
Ms. Ayana Forte, tell me why you decided to do this kind of research and what stimulated you to move and really ask this question as far as HIV is concerned.
Well, I definitely have an interest in mental health myself, as well as in neuroinflammation and neuroscience. However, this project came about because we realized that not a lot of studies focus on the connection between HIV and neuroinflammation and the effects that HIV can have on the brain. So we wanted to build more onto solving more issues other than just the initial infection.
Thank you.
Thank you.
Other questions? All right, thank you, Ayana. And there's a question from Audrey Lewis about having treatments for mental health combats the effects.
Can you repeat the question, please?
Audrey, are you able to elaborate on your question? It was a question in the chat box, and so we can follow up later.
Okay.
That question. Oh, so she's asking, can mental health assist with symptoms of HIV? And she joined the presentation late.
I can't exactly say if mental health can assist with symptoms of HIV, but in my understanding of it, it definitely can, because mental health is a huge aspect of HIV. As I mentioned, the diagnosis of HIV itself can be a form of a traumatic experience. So combating mental health and making sure that you're doing things to keep your mental health stable is definitely going to help with some of those symptoms of HIV, as far as the mental part of it.
Yeah, because you mentioned about post-traumatic stress disorder. Can you maybe elaborate a little bit on that from the research papers you read?
Like, what do you mean?
If you're able to. It was something about post-traumatic stress disorder.
Yes.
So that's a form of stress, and stress causing worse mental health.
Yes. The neuroinflammation that HIV causes is a form of oxidative stress. Oxidative stress causes the neuroinflammation that we see in HIV. And the connection is that post-traumatic stress disorder is a form of stress that also leads to neuroinflammation itself.
Ah, okay, got it. Thank you. Thanks for that explanation.
No problem.
That was an excellent presentation.
Thank you so much.
Thank you.
Thank you, Ayana. Our last presenter is Hayat Sabri Marshall.
Good afternoon, everyone. My name is Hayat Sabri. I'm a rising sophomore at Spelman College, currently majoring in chemistry. And this summer, I had the opportunity to work with Dr. Kimberly Williams, specifically focusing on a literature review on estrogen and neuroinflammation in an attempt to gain more insight into possible neuroprotection for traumatic brain injury. I originally got interested in this idea because I have a personal connection to TBI and thought it would be enlightening to research how estrogen may be playing a part in the neuroinflammation that occurs in traumatic brain injury. Traumatic brain injury is classified as an acquired insult to the brain by a mechanical force, and this can lead to temporary or permanent damage to the brain. In 2014, more than 2 million people in the United States suffered from TBI due to different insults. 28% of cases are from falls, 20% are from traffic accidents, 19% from direct impact to the head, 16% from unknown reasons, and 11% from assaults, along with a small percentage of other transportation accidents and suicide. Symptoms of TBI are usually classified into 4 categories, which depend on what area of the brain is afflicted. These categories are thinking and remembering, emotional or mood, physical, and sleep. So a patient who has just been diagnosed with a traumatic brain injury may experience difficulty thinking clearly, sadness, headache or nausea, and sleep irregularities. There are 2 phases of TBI, the primary phase and the secondary phase. The primary phase includes the mechanical disruption of brain tissue, which can lead to impaired blood flow, metabolic imbalance, tissue damage, and membrane permeability, which then causes inflammation in the brain and a loss of the blood-brain barrier, which leads to the secondary phase. The secondary phase develops over a longer period of time and includes oxidative stress and an inflammatory response, which leads to cell death. Our lab is currently interested in understanding how inflammation plays a role in the central nervous system during various insults, including traumatic brain injury, with the hope of alleviating neuroinflammation. So what exactly is neuroinflammation and how does it play a role in TBI? This is the inflammation of nerve tissue, and this can occur through external stressors like injury or infection. In this case, it will be injury. Short activation periods of neuroinflammation can be beneficial and aid in combating foreign substances in the CNS. This inflammatory response is mediated by microglial cells, which are the resident immune cells of the brain. This response is also mediated by an infiltration of immune cells from the body, such as macrophages and T cells. These microglial cells are in fact the macrophages of the brain that act as an immune cell defense by surveying the area for debris and infectious agents to maintain a healthy environment for the neurons. Now, these microglia are dynamic cells and they have 2 forms of activation. Pro-inflammatory, also known as classical activation, which leads to a release of pro-inflammatory cytokines and other neurotoxins causing neuronal damage and death. The second activation state is anti-inflammatory, also known as alternative action, leading to a release of anti-inflammatory cytokines, growth, and reparative factors. During traumatic brain injury in particular, increased microglial activation and pro-inflammatory cytokines are seen as an effect of the insult. So really understanding how these processes work is important in order to possibly reduce the neuroinflammation occurring in TBI. One way our lab is interested in reducing this neuroinflammation is through the endogenous use of estrogen signaling. So estrogen is a ligand-inducible transcription factor hormone. Estradiol is the most abundant form of estrogen that's found in the body, and it can bind to two classes of estrogen receptors known as the nuclear and membrane-bound receptors. Of the nuclear receptors are estrogen receptor alpha and beta. The GPER30 is classified as a membrane receptor. Other selective estrogen receptor modulators are synthetic molecules that have the ability to bind to various estrogen receptors throughout the body and act as estrogen agonists or antagonists. This summer, I focused on studies that involved estrogen's role in neuroinflammation. Estrogen is said to have neuroprotective effects because it promotes healthy metabolism, cerebral blood flow, synaptogenesis, which involves the formation of connections between neurons. And lastly, estrogen promotes the growth of neurons by mediating the anti-apoptosis genes. It is also speculated that estrogen receptors enhance this process, which may contribute to estrogen's neuroprotective effects and various neurological disorders, including traumatic brain injury. Previous literature has shown that early after TBI insult, estrogen levels are decreased in the blood by 7 days and may be reduced long-term up to 6 months. Interestingly, females have a lower mortality rate and less complications compared to males, and patients with higher levels of estrogen have been associated with lower mortality rates and a better Glasgow outcome score, which is used to measure the severity of TBI. Now, this score ranges from 1 to 8, with 1 being the worst outcome for the patient, death, and 8 signifying a prognosis for a good recovery. In rat models of TBI, neuroinflammation processes have been explored. In a study done by Chaudhry et al. Using a TBI model in rats, researchers were looking to observe the potential benefits of estrogen on traumatic brain injury and were able to see that estrogen reduced the edema in regions of the brain. Now, this TBI was induced in rats by a fluid pressure on the exposed dura after a connectomy, and subjects were injected with estrogen or a vehicle solution. Due to this induced TBI, edema was expressed in rats, which is a puffiness in the brain that's caused by excess fluid. So the edema area in this case is going to be indicated by the yellow arrow in the figure on the left. This edema was observed with a CT scan in the sham group, which is a group that underwent the same crenectomy but was not injected with the estrogen. The vehicle and estrogen sulfate treated groups were also observed. The figure on the right presents the averaged edema size of the groups on days 1 and 7. So if you really compare day 1 and 7 of the estrogen treated group to day 1 and 7 of the vehicle group, you can see that estrogen aided significantly in decreasing the edema size. In a similar study done by Kaksari et al., utilizing a TBI rat model in which they dropped a 250-gram weight on anesthetized rats, researchers really wanted to understand the role of estrogen in inflammation. So first they looked at the level of edema in rats, which can be seen right here. And they saw that the rats subjected to a TBI had increased edema compared to rats not induced with TBI. This increase in edema was significantly reduced when animals were treated with estradiol after TBI insult. This reduction caused by estrogen was reversed when estrogen receptors were blocked by the antagonist ICI. And the white bars in this case represent the vehicle-treated rats with the drug DMSO. And DMSO in this case was like an oil. Next, they wanted to assess brain levels of various pro-inflammatory and anti-inflammatory cytokines after traumatic brain injury insult. To do this, they looked at pro-inflammatory cytokine IL-1 beta and anti-inflammatory cytokine IL-10. So TBI insult increased IL-1 beta but decreased IL-10 compared to non-TBI rats. Treatment of estrogen reversed these effects, leading to a reduction in pro-inflammatory IL-1 beta and an increase in IL-10. This all suggests that estrogen reverses the neural inflammation that's seen during traumatic brain injury. Now, given all of this information, we can hypothesize that estrogen may be able to reduce the effects of traumatic brain injury. In the future, I would like to conduct an experiment using an animal model where we could induce traumatic brain injury by an advanced blast simulator, which means that participants will be exposed to compressed air on the scalp, and we would observe the effects of estrogen by injecting the subjects with this estrogen and specific receptor estrogen agonists or antagonists, excuse me, to explore each receptor's role in neural inflammation and TBI. Possible ways we could observe the effects of estrogen on TBI is by conducting behavioral tests, looking at learning and memory, or looking at the inflammatory markers or microglia activation in numbers. This summer, I was able to enhance my critical analysis, literature review, and research design skills. I also feel that I was able to really narrow down what kind of research I'd like to pursue in the future. Before I close out, I would like to thank Dr. Williams and the RISE program for the pleasure and opportunity to conduct research this summer. I would also like to thank my RISE sisters for supporting me throughout this process. Here are my references. Thank you for listening. I'm now opening the floor up to any questions or comments.
Good job. I had a quick question for you.
Yes, thank you.
Yeah, no problem. You said that this experience helped you kind of figure out like what areas of research you're interested in. Could you kind of like talk about what else you consider doing in the future?
Well, right now, I have a lot of interest. So for a while, I was interested in like chemical engineering and thinking about making hair care products and skincare products specifically for Black women. I also considered forensic science, but I really have an interest in neuroscience also. And I think that this summer really showed me that this is something that I'd like to pursue in the future.
Thank you.
Yes, thank you for your question.
I have a quick question for you, Hayat. First of all, great job. And then second, for your future research, are you leaning more towards an in vitro or in vivo model? Were you able to hear me?
I could hear you. Yeah, I heard you at the end. I would say right now, I think I would be leaning more towards an in vivo model just because all of the papers that I read this summer, even outside of the ones I presented today, have really focused on in vitro models. So I might consider in vivo, but I'm not positive right now. But thank you for your question. Are there any other questions?
I have a question. It was a brilliant presentation, Hayat. I think that we kind of rely a lot on like natural healing and like neuroplasticity after like a TBI. Do you think that based on like your literature review that there is any like it would be helpful to administer estrogen in the early stages of TBI more than the later stages?
Thank you for your question. And I think after my research this summer, I read a lot of papers where estrogen was administered in the early phases of traumatic brain injury. And I believe that the neuroprotective effects were heightened when this was administered in the early phases rather than in the later phases of TBI, where the neural inflammation could progress and cause these neurodegenerative effects. Thank you for that question. Any other questions before I close out? No? Okay, well, thank you guys for listening.
Thank you, Hayat. This concludes our student presentation portion of the symposium. And now we will move on to presentation of certificates. Mrs. Bird?
Okay. Congratulations, RISE Scholars. Your research presentations today were outstanding. Y'all did a great job. At this time, we would like to present you with certificates for your successful completion of the summer research program.
So, as y'all know, we have our RISE First students. So these are our students who started in the program in the first year in last fall, and then they completed this summer. So Aaliyah Brown, certificate of completion presented to Aaliyah Brown in recognition of your successful completion, to Victoria Eaton, Jaya Fowler, Noah Odell, and Hayat Sabri, and recognition of completion of the first program. So congratulations to you all. And then we have our upper class students, Vanessa Eaton, Ayana Forte, Tita Tambidu, and Jacqueline Willis. Congratulations to you all for excellent presentations as well, particularly in this time of, particularly in this time of COVID. And so you all did an excellent job, and we congratulate you for what you've been able to do this summer. All right, so at this time, we're going to move on to a special presentation from some of your Spelman sisters, some alumni that we have, who sent some words of wisdom and encouragement for you all.
We can't hear it.
Katana, can you restart that, please?
Hello, RISE Scholars. I'm Tiffany King Peoples, class of 2009. I hope that you all were able to make the most out of this virtual experience. I know that it wasn't at all what you expected, but you guys were not alone. My lab only recently returned to bench work after months of reading papers, writing grants, and thinking critically from home. So please know that thinking about science, writing about science, and just learning new concepts are just as important as doing the actual bench work. In fact, that's where all the new ideas happen. So happy symposium, finish strong, and good luck on your presentations.
Hey, my RISE sisters, my Spelman sisters. This is Simone Sawyer here. I graduated from Spelman class of 2017. I got my Master's of Public Health at Johns Hopkins Bloomberg School of Public Health, and I'll be starting a PhD program this fall at George Washington University, where I'll be doing a PhD in the social and behavioral sciences and public health. I'm just here today to encourage you all that you can do it. I hope all the videos that you're seeing of wonderful, brilliant, smart Black women that are doing stuff encourages you that you can do it too. I know there were times where I was sitting in you guys' seat and I was like, how am I going to make it? How is this going to work out? But I promise you, you are in the right place at the right time and things are going to work out for your good. So just know that Spelman is going to prepare you. The RISE program is going to prepare you. Utilize your support systems and make sure you have a purpose for the things that you're doing. Get you a higher power to believe in, to call on when things get weary. Ground yourself and knowing who you are and what you have to offer this world is enough. And don't forget to use your support systems. We love you. We got your back. And I can't wait to see all the great things that you all do. Please feel free to contact me anytime or any of the other RISE sisters. And we look forward to seeing you guys succeed.
Hi sisters, I am Janae Best and I graduated from Spelman in 2018 with a degree in psychology with a minor in public health. I just graduated from the University of Michigan School of Public Health with my master's in public health. And I'll be starting at the University of Michigan this upcoming fall with my PhD in public health, focusing on health behavior and health education. Trying to think of encouraging and inspiring words for you all. I know this is a really tough time for a lot of people. So I think first I would just say to try your best to remain undaunted. As Spelmanites, we know that, something like being undaunted is not new to us. We are resilient women. And with that being said, part of resilience is self-care and recognizing that self-care is a radical act. So being sure to find time to practice intentional self-care during times like this. I'm currently working on a lit review right now and doing research of my own. So for me, that looks like, taking breaks in between doing work, having really clear defined boundaries for my work. So like only working from like, let's say 9 to 5 and letting all my other time be like my free time, my self-care time. I would also say that, even though we are, in the midst of a pandemic, that the grind doesn't stop. And so if you can find ways to be productive and to find out, what really works for you, like what conditions do you need and how can you best create those conditions for yourself in times like this, that knowledge will take you super, super far. If you can figure out how to be productive now, that will help you in the long run to be able to accomplish whatever you'll need to be accomplished, whatever you will need to accomplish, whenever you'll need to accomplish it in the future. So yeah, those are just my 2 thoughts. I hope that's helpful.
Hey ladies, I'm Brianna Spivey, class of 2019. Currently, I'm a student, doctoral student at the University of Georgia in clinical psychology. And so I just wanted to leave you guys with some words of encouragement because I know right now we're all struggling, trying to stay encouraged and productive. So the first thing is extend yourself some grace. Be kind to yourself. Take time for yourself. Don't respond to those text messages in the moment or those emails. Take time to yourself. Second thing is also trusting in yourself, knowing that you are right where you need to be and where you should be. And so accepting that and knowing that there are no mistakes. You're where you need to be. And lastly, just being intentional with what you do with your time, what you don't do with your time, and how you protect yourself, always being intentional. So I hope you ladies, are encouraged by these words and the words of our sisters. And I hope you guys have a great year. Bye.
Hi everyone, my name is Gabriel Webb, class of 2019. I'm a second year PhD student at Harvard University. On my hardest days, I tell myself some things to help me through it all, and I'd like to share them with you. Firstly, you can do this. Everything you need to succeed, you already have. The work ethic, the faith, the potential, the intelligence. There's nothing anyone can do to take that away from you. Arm yourself with that and push on. If it was easy, everyone would do it. Next, practice self-care. For me, this means I make a matcha latte or a super sweet iced coffee, knit, go outside, learn one of Beyonce's dance routines and homecoming, whatever makes you smile. And lastly, ask for help. Are you confused? You're struggling? You need encouragement? Ask your friends, your family, your mentors, and your older RISE sisters for what you need. You can't do it all alone. You don't have to do any of this alone. We would love to help you. We're rooting for you and you're doing great.
Hello, ladies. I am Angel Bower, class of 2019, and a current PhD student at the University of Chicago. My first piece of advice is to please be gentle and kind with yourself. I know Spelman places a lot of pressure on us to always be our best, but you can't be your best if you're not practicing self-care. So please, please, please take time out for yourself. It's so important. And secondly, please remember that you lack nothing. You have everything that you need to succeed. Please don't second guess your abilities. And thirdly, you have a group of older RISE sisters who are ready and willing to help you with whatever we can. So please don't hesitate to reach out. We love you. We're rooting for you. Keep up the great work.
Hi everyone, I'm Ariel Blakey and I graduated from Spelman in 2019 and I'm currently a rising second year in the clinical psychology PhD program at Boston University. And I just want to congratulate you guys on a summer well done, on a job well done, and really encourage you guys to just continue to persevere and keep the faith and continue to have positive attitudes despite everything that's going on, because it's all been tricky and different and really unexpected, but I'm sure that you guys handled it with grace and with just an insane amount of tenacity. And I hope that you continue to, because the future really has so many great things in store for you guys. And I really, really am excited for you all. So best of luck, best wishes to you. I hope that you have a wonderful rest of your summer. And I'm just so proud of you all. Bye.
That was so nice. Thank you.
That was so nice.
Thank you. At this time, we'll have closing remarks by Dr. Bradley Brennan.
So this is going to date me, but I just marked 15 years with the RISE program at Spelman. And much of that time, I was a managing program director, interacting weekly with students. But since taking on this position in the administration as vice provost for faculty, I don't have the joy of being able to work with our RISE students week in and week out, which is something that I really, really miss. These presentations today were amazing. And congratulations to all our presenters. I loved hearing about what you've been working on for your research and what the program has meant for you and your plans for the future. As some of our graduates just alluded to, despite everything that has happened in the year of 2020, particularly this summer, you should be applauded for the fact that you kept your eyes on the prize. You worked so hard on your projects and your presentations that we enjoyed today. And that kind of perseverance and focus are exactly the traits that you'll need to pursue your research careers. We need your voices in science. We need you to be sitting at the table, to be leading the charge, and asking the questions that others are not asking. Thank you for letting us be a part of your journey. We'll do our best to continue to provide you with the experiences and the resources to support your success. On behalf of Dr. Blankson and myself, I want to also say a heartfelt thank you to our faculty research mentors. The dedication and effort that you show day in, day out, ensure that our students have an exceptional training experience. Dr. Blankson and I would also like to take this opportunity to thank the RISE staff, both former and present. You were able to pivot when we went virtual and you kept this train running. So well done, thank you. I think that our first virtual closing program of the summer was a great success. I also wanted to say how delighted we are to see former students join us. I saw many in the first hour that had to get off, and of course we had the beautiful video that we just enjoyed. Thank you for continuing to support the program. We love that you continue to be a part of this work, helping to train your Spelman sisters as they follow in your footsteps. We're so proud of all that you're doing, and we thank you for serving as role models for the next generation of Black women scientists from Spelman. Finally, thank you to everyone else who was able to join us today. I know that our presenters and all of our students appreciate your continued support. Now, if we were to face-to-face, this would be the time that we would invite everyone to a lovely reception or a lunch, but you're going to have to do that on your own today. Instead, I'll just send it over to Dr. Blankson.
Thank you for those words, Dr. Bradley Brennan. And this concludes our first RISE virtual summer symposium, but I don't think it'll be the last virtual. Even when we come face to face, I think inviting others in virtually was very excellent. And so thank you all for joining us, and hope you have an enjoyable rest of your summer. Bye.
Bye. Thank you so much.
You're welcome.
Thank you.
Thank you.
Bye, everybody.
Thank you.
Thank you so much.
Excellent.